So the next speaker is Sid from the University of Edinburgh, and he'll be talking about developing pipelines for transcription-wide association studies and applying them to some of the cohorts.
So it's just the green button.
Yeah, should be.
Hi, good morning. So I'm here today to talk about the work we did in detecting phenotype-associated genes by combining gene expression and GWAS summary statistics. And I would like to thank Emily and James for giving me an opportunity to work on this project, and our BOVREC partners to actually sharing the data which was needed to actually get the work done. So to begin with, I'm going to talk about TWAS, or transcriptome-wide association studies, which is kind of going forward from the GWAS studies which we have heard this morning. And what we're trying to do is, GWAS is trying to associate genomic regions with traits, and with transcriptomic TWAS analysis, what we're trying to associate is bridge the gap and say what genes are driving the traits. And this uses GWAS data and then combines that GWAS data with gene expression data to get to the point where we want to be. So why use TWAS? There are various points associated here. So we say it is kind of very tissue specific. It's also very efficient in predicting gene expression data, because we are kind of only associating genes to traits and not all millions of SNPs. So it's also faster. And it's also kind of giving a deeper understanding into the biology of the associations we are seeing. So it kind of taking it forward into how it fits into the whole BOVREC work packages, and it actually brings together work from different work package tasks. So for our analysis, we start from our TWAS cohorts, which is a group of a small set of samples where we know both the genotypes and the gene expression data. And then we can train models on these TWAS cohorts, and this is where we used data from the work package too. So the BOVREC annotation, which kind of greatly expands on the models, gene models, and the transcript models that are available. And once these gene models are generated, then we use data from the work package forwards of the GWAS data sets to kind of predict all the gene expression levels and then do the association studies. So to get this work done, what I did was generate two set of workflows, because early on we realized that there aren't many TWAS cohorts which we can actually directly work on. So what we decided to do was take RNA-seq data sets, which were large enough cohorts available, and predict genotype or impute genotype from the RNA-seq data sets that are available. So the first part of the workflow is just to generate the input files that are needed to train the models. And the second workflow kind of prioritizes on training the model itself. So I'll just go briefly through what these workflows are doing. So the first part of the workflow, like I said, is taking in the RNA-seq data and the annotation, like the BOVREC annotation, and then we are doing the gene expression analysis here. So star for getting the gene expression data, Callisto for transcriptomic analysis. We are generating the covariates using Peer tool, and then we are kind of imputing the genotype of these samples. And here we are using GLIMS instead of Beagle. What we saw was in imputing sites which are non-reference allele, GLIMS was performing better than Beagle. And once we have all these input files from the first workflow, it goes into the second workflow, where we are kind of using the same methodology it was used in human GTEx projects of training these models. And then all these workflows are in Nextflow format, so it can be tweaked, and it's good for reuse. And we heard a lot about Nextflow in the previous presentation, so it's on Nextflow. And then once these models are trained, they are kind of compatible with PredictXscan set of tools. So if you have used those tools here, so it's like a database file which you can directly plug into your PredictXscan analysis. So to get our TWAS model training going, what we used was like 5 tissue types. We took RNA-seq data sets from public data sets, as well as within our BOVREC partners, we used samples which were submitted in the BOVREC Ensemble portal. And what we did quantification using both ensemble as well as Bovreg annotation. And what we saw there was the reads which were falling into the gene features were significantly more in the new Bovreg annotation compared to the ensemble one. When we looked at the spread of the sample, the PCA plot on the right-hand side, all the tissue types were clustering as we would expect. The digestive tract tissues are together, and even the samples within the tissue types are segregating closer. So we are happy with this cohort, and we went ahead with model training. And as the models were trained, they do go through multiple filtering steps. So in the gene expression level, we just make sure that at least 50% of the samples in the cohort are expressing that gene. And once the model is trained, there is a filtering on the model parameters as well. So it's a z-core p-value less than 0.05 and a row average of 0.1. That kind of gives us a good quality models. It might be difficult to read on the slides, but on the bottom left side, it kind of gives the count on how many models we have for different tissue types. This is a summary for the gene, and it's for one of the GWAS cohort from ETH. And it's kind of representative of the other models as well. So on the gene sides, we get around 150 to 900 genes in our model. With the mammary gland and the digestive tract tissue having the highest amount of gene models, and the skeletal muscle the least amount, which kind of reflects on the type of transcriptomic activity in these tissue types. And looking at the subplots on the bottom right-hand side of the points, we see that there aren't many overlap or many gene models which are present across all tissue types, and the intersection is the highest where the tissue types are similar. So for example, the rumen and the jejunum has the highest intersection point there. But if you look at the transcriptomic models, we obviously capture more models. So on an average, we are getting between 1,000 to 4,000 transcript models across the tissue, with the overlap looking very similar to the gene models. So I did talk about the GWAS cohorts which were used. So I need to stress that as well. So while training the models, we kind of restricted the SNPs that are actually used in training the models to the GWAS cohorts themselves, so that we're making sure that the GWAS cohorts kind of matches the TWAS models, which we are testing it against. So we used the USDA GWAS trait, which is publicly available, and this was the set that was used in the cattle GTEx as well, which is a similar study in the same field, which has been already published. And then from Bovreg side, we have from our partners summary stats from ETH. We have the 11 dairy traits from brown Swiss cattle with around 5 to 12K sample size. Then from INRA, we have other set of cattle. It's kind of between Holstein and French breeds, and it is around 10K sample cohort size as well. And from FBN, so instead of taking the GWAS data, we actually sent our models to FBN. Praveen actually did the TWAS analysis on their side and then shared the data with us. So we have all these output results for TWAS hits from using our model on the Bovreg GWAS cohorts. I can't go through all the results, but I would like to share kind of representative of what we are seeing. So here in this particular figure, it is comparing protein percentage across in a mammary gland tissue using a USDA GWAS result. On the top half of the figure, we have the Bovreg TWAS hits. On the bottom half, we have the cattle GTEx TWAS. Like I said, it's a study previously done in the same field. And what we're trying to say is what are the kind of the hit overlaps. So the gray point is, the gray spots are the GWAS hits on the, across the chromosomes. The blue spots are the hits on our using the Bovreg TWAS, and the green yellow spots are the significant hits in the cattle GTEx side. And the cutoff is it's Barfoni corrected p-value cutoff there, and the points that are highlighted are the ones that are significant. And what we are saying is it's not that we are detecting the exactly same genes, but there is overlap, as well as we are detecting genes which are only present in our BOREG annotation side and not on the cattle GTEx side, and there are sites which are targeted, which are detected in the cattle GTEx, but not in ours. So it's complementary, and some of the sites that are highlighted, like PCAM and the Selenop, they're all kind of only detected in our BOREG side, and there are other novel transcripts which don't have annotation for, whereas some of the popular hits like DGAT1 is not present on our side. But then the reason why that happened is because we didn't actually have a significant model for those that chromosome 14 region in our mammary gland set, but we do have that region in other tissue types. And it is kind of taking point in what Gabriel said about the string type models. So for that region, it seems that all the different gene models are being combined into a single string type model, and that's why we didn't kind of capture a significant model for that region, for that tissue. But for the rest, we have it, and when we use the ensemble annotation, we do have models for mammary gland across this as well. So we do detect DGAT1 when we use the ensemble annotation. But we have with BOREG, of course, we are finding new sites which were not present in the ensemble. And if we kind of summarize the whole thing by traits and tissue types, we can actually see and compare what genes or what the hits are actually kind of present across different traits and which tissue types they are present in. So in these graphs, we are only showing those genes in the rows which are at least have one significant hit in one of the traits. And the traits are filtered so that we are only looking at those traits for which we have significant hits. And the colored strips are kind of showing which chromosome they fall into. So the sections are color-coded based on the chromosomes. So this kind of gives an idea of what we are trying to kind of capture with our TWAS analysis, and that should give us an insight going forward. And we are going to, as a conclusion, I would like to say that we are working towards the manuscript, and I will be putting all these workflows on GitHub, and it will be accessible publicly. And with that, I would like to thank all our partners, and thank you for sharing the GWAS summary data, and that kind of helps us with our analysis. Thank you very much.
Thanks a lot, Sid. Any questions for Sid? James?
James Coltis, Iowa State. Thanks for the interesting talk. One question regarding the comparison to GWAS results. Did you try or could you try using the livestock QTLDB? There are a lot more studies there, the ability to separate breeds by ontologies, traits by ontologies, group traits by ontologies, et cetera, to make comparisons.
Okay, so, thanks for the question. I haven't directly done it yet, but it's something we can look into. And in this case, because these were kind of trained, so the model was from that GWAS cohort, so it was easy to kind of overlap it, but there's something I can go and have a look, definitely. Thank you.
Any other questions? I'll cheekily ask you one, Sid. So we saw a lot of GWAS, really nice GWAS results presented earlier today. Yeah. So how long does it take if someone who did these GWAS, if they wanted to run their TWAS analysis using your models on their GWAS data, how long would it take them, do you think?
So there are two steps to it. So if you have your GWAS, all you need is the database file, but the only issue there is the GWAS ID, the SNP IDs have to match the SNP IDs and the current model. But if they already do, then it's just about running the S-predix scan. But like we said in the thing, we are actually training the model so that we only keep the SNPs in the GWAS studies. So they have to rerun the training model training step, for which we can provide the gene expression data and the SNP data, the genotype data from our cohort. So that's the second part of my workflow. So you just have to run the second part of my workflow, and then you can get the models and just run the predict scan. So timing-wise, it's just hours.
So they just need the summary statistics.
They just need the, yeah, of course. Well, it could be like full genotype level as well. So they just have to choose whether you use S-predict scan or predict scan.
Okay, thanks.
Peter.
So there's
So to come back on DGAT, do you have an explanation why you didn't find any signal? Is it due to the multiplicity of causal variants, or the splicing, or the fact that the major mutation is a coding in the coding sequence and affects the activity and not the regulation, or any explanation?
For the DGAT1 region?
Yes.
Oh, what we saw was in the string. So if we use ensemble ID, we do get DGAT1, and it was on our first deliverable. We have a list of genes, and DGAT1 is right there. At that time, we used the ensemble annotation. But when we used the Bovreg annotation, what we saw was the string the ID, the MSTRG ID, is actually combining two almost like a megabase region where DGAT1 is. So it spans the entire region. So when we are doing the expression, when we're kind of estimating the expression levels, it's kind of not there. The signal is not there, and then the model that is being trained is not significant enough in predicting the gene expression level given the genotypes. So it's getting muddied there, but we have to kind of tease it apart manually going forward. It's just the way the string time model is at the moment.
We know that in our breeds, for instance, so the French breeds you used, at least in one breed, the usual variance K232A is absent, and we still have the QTL there. Right. And so we know that there is a multiplicity of variants, and we don't know exactly which there are. And in addition, the region seems to be extremely difficult to impute, so also we probably have imputation errors.
Something to look into. Thank you.
okay. There's no other questions? None online? Okay, well, thank you very much.